Let's quickly review where DNA is found. Remember, DNA is found in your blood, but specifically your white blood cells. Your red blood cells do not have a nucleus, and so for us eukaryotic organisms, that means that our red blood cells do not have DNA. But our white blood cells, which have a nucleus, contain our DNA. We can also get DNA from bone, hair, preserved body parts, clothing and fibers, and saliva and other bodily fluids. And with that DNA, we can create what's called a DNA profile. Hopefully you read about DNA extraction. Don't forget that there is that extra credit opportunity to extract your DNA using common household materials and then take a picture. So now we're thinking about once we've collected and extracted that DNA, what do we do with it? So the first thing we have to do is we have to make lots of copies of it using a process called PCR, or polymerase chain reaction. That's going to allow us to have enough sample to perform a restriction digestion, or the breaking down of that DNA into smaller parts. And it's those smaller parts that are identifiable and act as our fingerprint. So restriction enzymes can break down our DNA at specific sequences. Here are some examples of some restriction enzymes, where they were originally discovered, their strain if it's known, and the order of discovery, which helps you figure out the name of that particular restriction enzyme. So EcoR1 is short for Escherichia coli, that's the ECO. RY13 becomes the R, and it was the first strain of that bacteria discovered, so EcoR1. Hi3 from the bacterial source, and it is the third one discovered. Hind3, similar, but it's Haemophilus influenzae, and it's the third one, so it has a different abbreviation. Each of these restriction enzymes has a specific recognition site that it identifies and will cut the DNA at. EcoR1 finds the sequence GAATTC and cuts between that G and that A. Hi3 finds GGCC and cuts down the middle. Hind3 finds AAGCTT and cuts between the A's. Now remember, some restriction enzymes cut down the middle, creating what we call blunt ends. Other restriction enzymes cut jagged, where it's not a smooth cut, it's not straight down the middle, and we call those sticky ends, because imagine like Velcro, some are shorter, some are longer, so they can stick together. So here we had an example of digesting with EcoR1. So you can see EcoR1 cuts at GAATTC between the G and the A. So first it identifies those sites, figures out where it needs to cut, and then cuts the DNA at those sequences. Based on the length of those sequences, we can run what's called gel electrophoresis, which you'll learn about in the next activity. So let's talk about your practice. So for your practice, let me see if I can make this bigger. For your practice, you are asked to digest the sequences using Hi3. We have to remember that Hi3 finds the sequence GGCC and digests in the middle. Technically, it finds GGCC on top, CCGG on the bottom, but if we know the top, we know the bottom because they are complementary. The only exception is if there's mutations, but we're not going to get into that. So when I'm copying down my practice problems here, all I need to copy down is this top strand of DNA. Now I know that I am digesting using Hi3. Remember, Hi3 finds the sequence GGCC and cuts down the middle. So first thing I'm going to do for my sequences is find everywhere that there is GGCC. In the first strand, starts at the very beginning. If I keep going, there's another GGCC here. Keep going. This is almost CGG. Oh, but there it is, GGCC. And if I keep looking, GCCG. Oh, nope, that won't work. So this will digest 3 times. And what I mean by that is there will be a cut here, a cut here, and a cut here. So there will actually be 1, 2, 3, 4 fragments of DNA produced. To count those fragment lengths, we use the top strand and we literally just count the bases in each fragment. So this would be GG, that's 2, CCTCGG, that's 6 base pairs. We abbreviate that BP. A lot of times you'll also say CKBP for kilobase pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 base pairs. 1, 2, 3, 4, 5, 6, 7, 8 base pairs in that one. So we'll digest this second strand using the same method because we're still using high 3. So we're looking for GGCC. GGCC. That's the only spot where I see GGCC in the top strand. If you're looking at the bottom strand, you're technically looking for the palindrome or the opposite. So like here, GGCC would be the match, but I'm not looking for GGCC in the bottom. I'm only looking for it in the top. If I was looking at the bottom, I would need to find the complementary, which is that CCGG if we're reading left to right. So I'm going to go ahead and digest this, cut between the G's and the C's, count my base pair lengths, 1, 2, 3, 4, 5, 6 base pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21 base pairs. And you are going to try doing something like this on your next practice sheet. You will have to look up some different restriction enzymes. I like to do that by looking up, say, the image of it. So for example, if I want to say like HIND-3 digestion enzyme, I like to look at images and I verify that this is HIND-3. I see that this is HIND-3. It cuts between A and A in the sequence A, A, G, C, T, T. So I'm going to look for this sequence. It is not an even cut. It's not straight down the middle. So this would produce blunt ends. And then if this is what I was using to cut, I would find that sequence just like we did and cut accordingly. Again, you will apply that to the next activity. So there are 2 questions to think about before you finish activity 1.1.5. If DNA has 2 strands and the sequence of one strand is known, is it possible to determine the sequence of the second? Essentially, that's saying if you knew a sequence CTGAGGCCTA, could you tell me what that second strand is and how do you know? So think about the notes that we did on Thursday and Friday. If I gave you that sequence, what would the complementary sequence be and how do you know? The bottom question, can you be framed by your own DNA? How might your own cells lead to false verdicts of guilt? Provide a scenario in which this might occur. So think about the examples we've discussed in class. If a crime were to happen in our room, whose DNA would we find? And how could that possibly lead to false verdicts of guilt? If you have questions, we'll discuss more tomorrow.